Good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled Facilitating Target Identification Processes with AI, Challenges and Prospects. My name is Sonia Hunt, and it's my pleasure to be your X Talks moderator for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speaker. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speaker throughout this presentation by using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located in the control panel, and that's found on the right-hand side of your screen. If you require assistance, please contact me at any time by sending me a message using that chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available to you for future streaming on xtalks.com. At this point, I'd like to thank Standign, who developed the content for this presentation. Standign is a workflow AI-driven drug discovery company headquartered in Seoul, South Korea, and subsidized in the US and UK. Standign has proprietary AI platforms encompassing novel target identification to compound design to generate commercially valuable drug pipelines. Founded in 2015, Standigm has established an early-stage drug discovery workflow AI to generate multiple first-in-class compounds within 7 months. Pursuing full-stack AI-driven industrializing drug discovery, Standigm has achieved the automation of molecular design workflow, and the automation effort has been expanding to the whole drug discovery process based on Standigm's AI platforms, including Standigm Ask for novel target discovery and Standigm Best for novel compound generation. Now it's my pleasure to introduce you to your speaker for today's webinar. As a Vice President of Strategic Alliances of Standigm UK, Dr. Heejung Koo is focusing on establishing diverse collaborations regarding novel target identification and validation based on Standigm's proprietary AI platform for target research. Prior to Standigm UK, Heejung was leading the bio-platform team at Standigm Inc., contributing the conceptual design and application of AI-based target identification approaches at the biology domain in order to enhance and accelerate drug discovery. She received a PhD in systems biology from Pohang University of Science and Technology, and Heejung is the co-author of several publications about AI target research. Now it's my pleasure to pass over the mic and the controls over to Heejung. So Heejung, when you're ready, you may begin.
Okay, great. Thanks for the very kind introduction, Sonia. And I think now all of you can see my screen. Okay, so I'm really happy to have this opportunity to share my experience with you all regarding AI-assisted target identification. So as introduced, I work for an AI drug discovery company named Standign, of which the headquarters is located in Seoul, South Korea, and we recently opened a UK subsidiary in Cambridge. So I'm gonna give you a bit more of the information about our company to help you understand what we have been doing by giving you some background knowledge. Standigm is an AI drug discovery company, especially focusing on the early stages of drug discovery. The company is established in 2015, and now we have 76 members. We are a very well-balanced team in terms of expertise. The chemists, biologists, AI scientists, and platform engineers working together. Most members are working in Seoul, South Korea, in our headquarters. And recently, we opened international offices, first in Cambridge, UK last September, and then in Boston, US as well. We have an experienced executive with different backgrounds. These are our three co-founders with expertise in AI, systems biology, and systems engineering. And our directors are specialized in machine learning, biology, and chemistry. And these are our advisors, one for business development and two for AI drug discovery and algorithm. Especially Dr. Namjik Han is helping us also in establishing global partnerships. This is a simplified figure showing the concept of our workflow. We have constructed our own workflow to get first-in-class compounds from novel target identification. So these two are our representative AI technologies. Standigm asks for novel target identification and Standigm Best for novel compound design. We focus on small molecule drugs at the moment. So to synthesize the design structured, we recently set up our synthesis lab in Korea, and we also work closely with CROs. For compound testing, we are making the most of the advantages of good collaborations with academia, pharma companies, and CROs. We keep trying to improve this workflow, shortening the overall time scale, which is about 7 months to 1 year, more or less now, making the success rate higher by improving our technologies for the very early stages. So this part is what I'm going to tell you today, AI-aided target identification. And with the workflow I explained in the previous slide, we've got some assets. So some are ongoing and some are ready for licensing. So I want to make it clear that Standign is basically a pharmaceutical company with strength in AI running its own pipelines. Now let's move on to today's topic. Before I start, I think it's good to briefly look into the current situation that we are facing to make sure we're all on the same page. But let me be quick because I don't want to take up too much of our time here. One of the biggest obstacles we have in drug discovery and development is obviously selecting the right target. Considering more than half of the failures are because of poor efficacy, as you can see here, actually, I don't even want to imagine the situation that what if we have been developing therapeutics for the wrong target? It's definitely a nightmare, wasting resources. But unfortunately, these things happen. So we can't emphasize enough the importance of target identification. And surprisingly, only 3% of human proteins are currently associated with drug mechanisms of action. This is the target categorization by Burroughs. T-clean here used as drug target. T-chem, there are some reported bioactivities. And T-bio, at least we know the function, and T-dark, a little information. So it's sometimes amazing and sometimes overwhelming that we still have a long way to go. And it is imperative to try and prepare some smart methodologies to make the most of our resources, such as the accumulated knowledge and data. So this summarizes our effort for target identification so far. I'll walk you through each approach and share some related issues. Working on this target identification things, what we should remind is that there's no one almighty method to solve this problem. Understanding the disease biology with the flooding data is quite tricky because different diseases show different aspects, and this cannot be interpreted with any single type of data. We need to understand what kind of information we can get from this type of data, and if there's any other information, we need to solve the problem better. And another important point is that the result should be accompanied by relevant evidence. However, I would say this is where a kind of compensation should come in, because imagine that a promising novel target with full of information enough to be explained that no one started to research. It's like a unicorn. I don't think we can easily get that. So we should understand how novel the target should be and what would be the appropriate information to achieve that degree of novelty. With these basic ideas, we're still in the middle of our journey, developing and improving 4 different approaches so far based on this diverse data sources. Each approach adopts different methodologies. Deep learning-based target recommender with this knowledge graph, and the natural language processing technology. This is the one using the omics data, and this is to predict the genotype-phenotype prediction. So these approaches are used to prioritize target candidates, and then we add some more criteria and information such as novelty, target safety, and market status to finally select the target candidates. Then it's time to go deeper into each approach. At first, we imagined how to improve the conventional ways to find novel targets. Researchers try to put the valuable information together as much as possible and try to find something new by integrating the information they've got. So this is why we decided to construct a knowledge graph, which is comprehensive, automated, and highly flexible. The graph looks like this, quite heterogeneous, including phenotypes, targets, drugs, and other related concepts, and the relations connecting the different node types. The heterogeneous information was collected from public databases and literature and curated by domain experts. Now we have about 4 million edges connecting about 70,000 nodes, and it's being updated continuously. We prepared the system for automated updates to minimize human intervention, and most importantly, the graph is flexible enough to accommodate external data, and thus it can be easily modified and improved. So when we do the collaboration with this system, we use a customized version integrating our collaborative data. Constructing the knowledge graph, what we tried first is to integrate the curated information from the available databases. However, obviously there was a limitation because we couldn't quickly update the most recent information from the publications. This is the reason we decided to develop a separate NLP engine to make the unstructured information structured. So this is how it works. Our NLP engine first recognizes the entities, the biological terms such as genes, diseases, and some other physiological activities, and relational words explaining the context. Then it predicts the relations between the recognized entities, so the unstructured information can be transformed into the structured information. Like this, you can easily find which concept is connected to another concept or how they are connected. Using this technology, we can not only keep our knowledge graph up to date, but we can also provide the summarized information to the researchers to help them review the target candidates with relevant information. We applied deep learning algorithm on our knowledge graph to understand loads of knowledge using machine intelligence. First, we use topological information of the nodes in the graph, such as the localization and local connectivity. These will be used as node features. And then we extract the path connecting the disease and target nodes. So suppose that this is a disease and a target, and there might be many paths connecting the disease and target, but it doesn't involve every single path existing in the knowledge graph. Normally, there are a lot of paths connecting a certain pair of disease and target directly or indirectly. But among all the paths, the algorithm suggests important paths that significantly contribute to the disease target association, and it can also decide which path contributes how much to explain the association. So this will work as path features, and by combining the node and path features, we can calculate the association score, and we can get a prioritized target list according to the scores. Additionally, the important path selected by the algorithm can work as evidence of prediction because they contributed to quantifying the associations. So we constructed a platform for internal research and visualized paths on our platform to facilitate further analysis. Simply speaking, the concept is like this. So let's suppose this is the knowledge graph. This red one is a query disease, and the blue one is a certain target. The algorithm will select important paths to explain the disease target association, and the set of important paths would be different for each target, of course. So we can get diverse information from this subgraph by analyzing the individual paths, and also we can use them to further narrow down the target candidates. So as we already know, we can't cover all the aspects of a certain disease, and we may have specific pathways in mind, or we may want to find some targets specifically related to our expertise and validate them with well set up assays. In these cases, we can narrow down the target candidates using the subgraph. I mean, let's say there's a pathway we are interested in, and then we can define a gene set related to the specific pathway. And then we investigate each subgraph with the predefined gene list. So if we already got 3 target candidates, as here, A, B, and C for the query disease, and the subgraph of the targets A and C contained one or more nodes from the previously defined gene set, then we can narrow down the candidate to A and C rather than considering B altogether. So it is helpful not only for narrowing down the number of candidates, but for obtaining targets more focused on our interests. So this is how the system looks like. We have constructed a platform to facilitate the analysis of prioritization results. We get the rank based on the association scores, and there is additional useful information such as gene dependency, gene functions, or development status. And this system also shows the visualized subgraph like this. To show you a bit more of the details, the platform provides the information about the relative contribution of each path. So this is overall subgraph, and when you move the mouse on the path information, the path is highlighted like this, and the users can modulate the graph by deleting or adding certain graph components so they can freely explore the whole knowledge graph, not just a provided subgraph. Also, the system provides fundamental information of each graph component, and the sources of information are also clarified. This is the system constructed for internal users for internal research, but we're also doing collaborations to customize this system upon our collaborators' request. With this approach, we've been doing some collaborations to identify novel targets for different indications. Recently, we've got some preliminary results from our collaborators. The first one is a case of fibrotic disease. So here, Standign suggested 28 target candidates using our system, considering the collaborators' research interests and experimental capacity, and they selected 3 of them after reviewing all the information we provided. They wanted to check whether the expression level of a certain fibrotic gene is altered. So they knocked down the target genes in cells, and all 3 substantially affected the fibrotic gene expression. So in this case, our collaborators are now working on the follow-up experiments. And this is another example of another fibrotic disease. Here we suggested 19 candidates, and 5 of them showed a reliable and reproducible effect on decreasing fibrotic gene expression levels when the genes were knocked down in the system. Here I brought only a part of the results, but you can see the candidate number 4 affects the fibrotic gene expression for all the cases compared to the disease induction condition. Among the 5 candidates with positive results, we decided on one to develop further. So this is also on the further experiment. As mentioned earlier, we're still in the middle of our journey, and there are some takeaways that we learned from our experience so far. I would like to summarize the key points and share them with you here. We definitely found that a well-constructed knowledge graph can be a good way to explore the huge space of information. We can expect a kind of emergent property from the comprehensive collection of useful information when they are properly connected. Of course, the type of information in the graph should be carefully selected according to the problem we want to solve, because otherwise, the algorithm may suggest a path that it understands to be the best to explain the relationship, but you may end up finding nothing from the past, even though you go through every single component of the subgraph. Given that the graph is properly designed to provide informative paths, they can provide different insights regarding the disease target associations. However, this cannot be the almighty solution, of course. So we could name some other points that we needed to improve our system. First of all, disease-specific information should be used to get more promising targets. For example, our knowledge graph had the general information about target expression at first. I mean, the general expression profile of target. So it was quite helpful to consider the general safety of the target, but it was not that useful, to be honest, to investigate how the target is expressed in the patients. In this case, we could construct different graphs for different indications or add additional filters to apply the corresponding information afterwards. Also, we found that this system could be improved if accompanied by different approaches with different data types and levels. So I'm going to talk about this a bit later. External data sources providing the information such as market status are essential as well. Finally, and most importantly, target candidates from the in silico approaches should be properly validated to make the target candidate meaningful for further development and to evaluate the system itself, and also to get the feedback from the validation results and improve the system accordingly. To achieve this, the experimental schemes for target validation should be carefully designed, and the system also should be able to provide appropriate information to design the validation scheme. So from now, I'm going to talk a little more about the different approaches and the validation plans. With the approach mentioned previously, we were able to deal with the knowledge level information quite well, but we felt there's still a limitation because the knowledge level information just allowed us to utilize well-curated, discrete pieces of information. Even though we have integrated the omics data into our knowledge graph, anyway, it had to be once translated into relations according to the predefined criteria, such as upregulated or downregulated. However, in terms of unbiased omics data, we believe AI can add great value to our understanding by interpreting the raw data with distinctive insights. So we try to find out how we can expand our method to understand the unbiased data. We started by applying transcriptome data to the protein-protein interaction network. The PPI network is a part of our knowledge graph, but it is slightly modified here. In a conventional PPI network, all the edges are treated equally. However, we can see that it may not work in that way in reality because some of the proteins are more tightly connected, working on the same biological process, and some interactions may be stronger or weaker according to the biological context. Considering this point, we assign different weights to the edges using the intrinsic network structure and disease-specific information. Once we obtain the weighted PPI network, information from disease transcriptome data is aligned on the modified network, and finally, we can get target prioritization scores by applying guided random walk. This approach contains roughly 2 steps, and the first step is to make a weighted PPI network. As I said in the previous slide, 2 things are considered here. One is network structure to discriminate the interactions inside and outside the modules. So here I mean by modules, a kind of protein complexes or some proteins involved in the same biological processes. So there are some interactions inside the modules, and there might be the interactions breaching the modules. And the other is disease-specific information that is previously known targets of the disease of interest. Therefore, the edge weight is given based on the previous knowledge, making the information tend to flow towards current knowledge. And because of this disease-specific information, the resulting weighted network is also disease-specific. Once we obtain a weighted PPI network, the next step is to apply transcriptome data to the modified network. Specifically, we calculate the amount of differential expression between disease and normal samples and apply the values to the corresponding nodes. It is quite unbiased information, and of course, it may contain noises inevitably. Here, we expect the dispersion of the input data through the guided random walk based on the edge weight can compensate for the intrinsic noises while maintaining the real unbiased signals in the data. To this point, we have been accommodating knowledge level data and transcriptome data in our methodologies, and the next action we focused on was introducing genetic information. Genetic information has been highlighted as promising evidence in terms of target identification, as targets with sound genetic evidence turned out to have a much higher success rate. We focused on a genome scale metaboling model based on our systems biology expertise. We expect the model to enable us to predict genotype-phenotype relations, especially when it comes to cancers. So this is what we're doing. First, we have constructed our own genome scale metaboling model. So it works as a basic scaffold comprised of all known reactions in human metabolism. Based on the scaffold, we can apply transcriptome data to make it context-specific. This is highly versatile because we can make a metabolic model with any biological context if we have corresponding transcriptome data. Here, we use cancer data from TCGA and control from GTX to set up 33 cancer-specific models and the same number of control models. The strength of each reaction can be quantified as flux values. So each model has a different set of flux values reflecting the relative strength of reactions in the different biological contexts. It is quite straightforward that we can obtain some cancer targets by comparing the cancer-specific and control models. Suppose that there are two different pathways to synthesize a certain essential metabolite. One is specifically used in controls, and the other is specifically used in cancers. Then, the enzyme mediating the cancer-specific reaction can be a potential anti-cancer target, and we can estimate how the overall flux values will be affected if we make the potential target disabled. This is one of the advantages of these genome scale models, that we can simulate the effect of one or more knockouts. In this figure, this would be the ideal situation that no normal flux is influenced by the target knockout, but cancers are greatly affected, which means a selective growth inhibition on cancer cells. So far, this is kind of a simulation tool, but AI can come in as well. In this example, using the flux values and the expected phenotypes from the simulation, we can train the model with the flux values as input and the cell growth rate as output. The sequence information can be reflected to the flux values by controlling the range of corresponding flux values. So the upside of this approach is that we can see which reaction is affecting the phenotype, because we can observe the overall flux balance, and therefore it can endow this approach with explainability. So we look forward to identifying pivotal SNPs for the phenotype with this model. And we have identified tens of cancer targets using this approach, and our collaborator is validating the candidates. So this is our journey, which is still ongoing. And I would say that we have equipped with methodologies using knowledge level and unbiased data, both generalist and disease-specific ones, the specialists. So we're utilizing these approaches internally or in collaboration to find novel targets and also establish a smart system for novel target identification. I already touched simple examples of our target validation efforts, and now let me summarize the overall strategy for our target validation plans. Currently, we are examining different methodologies for target identification by utilizing our modules separately or in combination to understand the characteristics and effectiveness of each module. Now we're testing 3 methodologies. First, using the deep learning-based recommender and the NLP-NG. We select the target candidates using the priority rank from the recommender and the information extracted by the NLP system. Target candidates for fibrotic disease and a CNS disease were selected with this methodology. For this fibrotic disease, our collaborator is a pharmaceutical company in Korea. They finalized in vitro assays for 19 candidates, and 5 showed a significant effect on fibrotic gene expression when knocked down. And now we are designing drugs for one target, one of the 5 validated. The second one also uses the deep learning-based recommender, but sorry, but more focuses on the evidence path from the knowledge graph. In case we are interested in a specific mechanism of action or pathway of a disease, we further investigate the supporting evidence from the recommender. And because the evidence is composed of the paths that significantly contributed to relating the disease and target, we can select the targets more likely to be related to a specific pathway. With this methodology, we prepared another fibrotic disease target candidates, and our collaborator has found that 3 out of 3 tested candidates induced substantial alteration of fibrotic gene expression when knocked down. So they are also under further experiments. So these two are the slides that I showed you previously. And the last one is to obtain the target candidates for a specific type of cancer using the genome scale metabolic model. Here we use the transcriptome and genome data of a specific cancer type from TCGA, and now the in vitro assays are ongoing. In addition to the current effort for novel target identification and validation, we're planning to expand our scope in this area. To resolve current limitations, especially our UK subsidiary is actively making new collaborations and preparing to hire people here, and we anticipate we can tackle the problems we're facing by the solid alliances and internal members, advisors, solely organized for target ID and validation. So these are the points we want to sort out at the moment. We need to secure proprietary data sources to get optimally defined and generated data to get the best results from our system. And sometimes our validation plans are discouraged because there's no available assay. Definitely, we need expertise in each disease area and also human resources for dedicated experimental design and assay development for validation. And then we believe we can improve our algorithms and systems much faster and in a more efficient way with the feedback from the validation results. So resulting in a positive loop of better systems and better outputs. So these are the collaborations currently discussed and ongoing through our UK office. The first one is a 3-way collaboration with university and a hospital for cancer target ID and validation. Standign is using its genome scale model to find the target for a specific type of cancer. And the hospital provides the data and the university contributes to data analysis. And for metabolic disease, this collaboration involves target identification and target validation and platform licensing as well. So this is quite interesting. So we have our own database and our own algorithm. So we've got our system, but another company has its own database. So they provide their database and we apply our algorithm to their database. So this is kind of building a customized platform for our collaborator. And with this customized approach, Standign will suggest target candidates for metabolic disease and the partner will validate them. So this is kind of pilot study, and then we can discuss for further collaboration. Another cancer target ID and validation collaboration. So we are partnering with some collaborators providing the omics data as access. The Standign will get the data from the partners and apply the data to some of our models, and the university will take part in the omics data analysis and target validation. And finally, we are discussing the automated and scalable experimental setup with a company, first for cancer targets. Basically, in this collaboration, Standign will provide a target list and the company will do the experiment. So I'm quite excited to discuss the collaboration scenarios, and some of them are already ongoing. So hopefully we'll be able to see some meaningful results shortly. So this is all I prepared for today. So thank you for your attention. I'll be more than happy to take questions.
Well, thank you very much, Heejung, for that very insightful presentation. I hope everyone enjoyed it. And just like Heejung said, we're going to now start the Q&A portion of the webinar. So we're going to put on our webcam. So Heejung, you can put on yours. And I have a bunch of questions that have come in while you were speaking. So I'm going to start with those. So remember, audience members, if you would like to send in your questions or continue to send in your questions, please go ahead and use the questions window that's located on the right-hand side of your screen, and we'll try to attend to them during the time that we have together. Okay, Heejung, here's our first question. Now, this member is asking, in the audience is asking, how do you validate the target candidates? Do you have your own wet lab? It's an interesting one.
Yes, thanks very much to ask this question. So we don't have wet lab, and we're not planning to set up our wet lab at the moment. And our plan is to make the most of good collaborations. And we're trying to build a good relationship with partners who can discuss systematic automated target validation at scale with us. However, we are also organizing an internal team dedicated for target validation to define the overall validation scheme and analyze the validation results to apply what we learned from the validation steps to improve our system. So we have our validation team, but we don't do the experiment ourselves, but with our collaborators.
Okay, perfect. Thank you so much for explaining that. Let's go on to our next question. Okay, someone in the audience has sent this question in, and they're asking, are the target candidates from your approaches all novel?
Oh, yes, it's a great question. The target novelty is an important but very tricky issue, as I mentioned during the presentation. And I would say the target candidates from our system are all novel because our ultimate goal is to obtain first-in-class drugs, novel target, novel structure. That, as I mentioned earlier, we need to define novelty first. In our case, first, we remove all the targets whose ligands have ever entered the clinical trials of the disease of interest. I mean, so if someone already did some clinical trials with this target and the target is not novel anymore, so we remove all those targets and then we quantify the novelty using our NLP and gene based on the co-occurrence of the disease and target in literature. And also we provide the summary of the known information regarding target using our NLP again. Aggregating all this information, we select target for further development with some clues of disease association, but with no developed drugs.
Okay, thank you for clarifying that, Heejung. Let's go to our next question. Here it is. You mentioned different data types during your talk, and you're using the diverse types of data to identify novel targets. What do you think is the most critical data type, or rather most critical data type is?
Yeah, thanks for the great difficult question. It's very hard to pinpoint one critical data type, but generally targets with sound genetic evidence are known to be quite successful compared to targets with other types of evidence now. However, maybe the best way would be using diverse types of data and trying to analyze diverse aspects of the disease reflected in diverse data.
Okay, thank you so much for that. Let's go to our next question. We have someone in the audience who is asking, how do you utilize the results, the information obtained from different approaches, and do you get similar ranks of prioritized targets if you try different approaches for a certain disease?
Yes, that's a very important question.
Mhm.
Actually, we get quite different ranks from different approaches, and I think it's natural because each approach would utilize and analyze different aspects of the disease. So the easiest way to collect the results all together might be just aggregating the ranks, but at the end of the day, I think we should be able to understand the approaches better and find out which one works well for which indication. Because as I keep talking, um, there's no one almighty method. So this method might work well with this indication, and the other one may work well with another indication. So we need to understand our approaches better, and then um, we can use uh, the suitable approach, the most suitable approach for this indication. So this is also one of the reasons that we are aiming for the systematic validation.
Okay, thank you so much, Heejung, for that. Let's go to our next question. Okay, we have a person in the audience that is asking, how do you decide what kind of information you would put into your knowledge graph? Is there any way of making a decision on what is proper information to integrate into the database to get the results you want?
Yes, this is a great question.
Mhm.
Basically, our internal experts, biologists and systems biologists, discuss what kind of information we should use and where to get that information. But we have an in silico measure to evaluate the quality of information. So we use some matrix to quantify how well our algorithm recognizes the previously known targets and try to make the system have higher matrix values. So this is based on the hypothesis that
Mhm.
if the system recognizes already known targets well, then we can also trust a novel target with high ranks that we have never seen before with this indication. So we decide first with our expertise and evaluate the results using the matrix and then make a final decision.
Okay, thank you so much for explaining that. Okay, we'll go on to our next question. This looks like it may be our last question that we're going to answer for today. In the audience, we have a member who's asking, what is your plan after validating the targets? Do you also design drugs for the targets?
Um, yes, our ultimate goal is to design and develop novel therapeutics for novel targets. So currently, we are focusing on small molecule um, design internally. So this system can provide novel targets to our internal drug design part. And we're also doing collaborations using this system. So we identify targets and our collaborators validate the target candidates, and the drug design can be done either by us or by the collaborators. It depends on the contract or the scenarios.
Okay, perfect. Thank you so much, Heejung, for answering all those questions. Well, it looks like that's it for our Q&A portion of the webinar. We've come to the end of that part. If we couldn't attend to your questions, the team at Standign may follow up with you after this presentation. And if you have any further questions, let me just change the screen here. There you go. There is Heejung's email address. So please go ahead and utilize it. She'd be more than happy to assist you with any of your inquiries. So thank you very much, everyone, for participating in today's webinar. You will be receiving a follow-up email from Xtalks with access to the recorded archive of this event. A survey window will be popping up on your screen. Your participation is much appreciated as it will help us to approve our further webinars. Now, give me a few seconds. I'm going to send you a link in your chat box, and you'll be able to view the recording of this event with that link and also share the link with your colleagues once they register for the recording as well. So I encourage you to do that. Now, please join us in thanking our speaker, Heejung, for that very insightful presentation. So thank you very much, Heejung.
Thank you.
Okay, we hope you found this webinar informative, there's something else I'd like to add. Heejung will be attending the 2022 Bio International in San Diego to introduce Standigm's target identification technology. So if you're interested in the technology, connect with her at that event, or please go ahead and utilize her email address. Okay, so it has been my pleasure to be your webinar moderator for this event. On behalf of the team here at X Talks, I'm Sonia Hunt. Please join us again. Until next time, please take care and bye for now. Bye, everyone. Thank you, Heejung. Bye, everyone.
Thank you. Bye.
Bye.